Hello, my name is Mohit Dalwadi, and I'm going to talk to you about a mathematical model for single-cell cryopreservation. The main goals of this work are to try and understand and quantify how different freezing protocols affect the survival rates of different types of cell. To go about this, we're going to develop a computationally efficient model of the process. The main ideas behind the quantification of cell damage in our model come from the two-factor hypothesis of freezing injury introduced by Mazur and co-authors in the 70s. This hypothesis comes from the observation that there's an optimal cooling rate to maximise cell survival for a given cell type, as can be seen from this figure. You can also see that different cell types have different optimal cooling rates. The two-factor hypothesis says that if you cool too fast, the cell is damaged by intracellular ice formation, which can pierce and crush the cell membrane, whereas if you cool too slowly, the cell is damaged by solution effects injury. For example, the toxic effect of cryoprotective agents, or CPAs, added to lower the freezing temperature. These two damage mechanisms are more prevalent at different extreme cooling rates, and so there's an optimal cooling rate somewhere in the middle. It's this optimal cooling rate that we want to try and predict using mathematical modelling. Since this is a cryobiology conference, I'm going to say a few words on mathematical modelling in general before discussing the model we propose. So, what is a mathematical model? At its most basic, it is a process by which one turns information into a prediction. A very simple example of this is a process you often make on whether or not to carry an umbrella with you when you leave the house. You look outside, take in information about the sky, and make a prediction about the weather. If you live in the UK, like me, you generally find that you should take an umbrella with you on most days. Models can, and often do, get more complicated than simply looking at the sky. The more complicated a model is, the more difficult it tends to be to relate the results of the model to the physical processes involved. Additionally, more complicated models tend to be much more difficult to solve. The key in modelling is to understand just how complicated you should make the model in order for it to say something useful, without making it too complicated and not understanding what the results of the model are telling you. The sweet spot completely depends on the application and what you're trying to learn. So, to get back to cryopreservation, what do we want from a model? Well, in order to understand the damage caused by ice formation and solution effects injury, we'll need to understand how the heat and CPA are transported through the system, and how these affect one another. We'll also need to track an advancing freezing front, and any growth or shrinkage of the cell. There's a really excellent trilogy of papers on the mathematical modelling of cryopreservation by Anderson, Benson and Kearsley, with the last two papers published recently. I'd really recommend that anyone interested in modelling cryobiological processes read these papers, as they are very comprehensive, fully setting out how one can go about modelling the processes involved in cryobiology and the challenges that this modelling entails. In terms of the mathematical model I'm going to talk about, essentially it involves a spherical cell inside a spherical domain of liquid culture medium that contains CPAs and other chemicals. We're going to cool the system from the outside, and this will cause ice to form and to propagate into the domain. We're going to say that the ice is pure and contains no CPA, so the CPA in the system gets more concentrated as the ice forms. This will, in turn, make the cell shrink due to osmotic effects. The heat transport is dominated by conduction and needs to be solved for in the cell, liquid and ice domains. Mathematically, this corresponds to these partial differential equations. We assume that the cell and liquid domains are the same in terms of the heat transport. The transport of CPA is dominated by diffusion, and this only needs to be solved for in the cell and liquid domains. This is because we assume that the ice is pure and contains no CPA. Mathematically, this corresponds to these partial differential equations. We need boundary conditions to couple these regions together. At the external boundary, we impose the temperature. At the freezing front, we have several conditions. The first condition says that the freezing temperature depends on the CPA concentration. A higher level of CPA corresponds to a lower freezing temperature. The second condition accounts for the latent heat released upon freezing. The third condition says that no CPA is captured within the ice. The ice is purely water molecules. At the final boundary, the cell membrane, we have these conditions. The first condition says that the CPA flux is conserved across the membrane. The second condition says that this flux is proportional to the concentration difference across the membrane. And the third says that the cell shrinkage is down to osmotic effects, so it's proportional to the concentration difference across the membrane. While there are a lot of equations and conditions here, the two most important for understanding what's going on are these two highlighted in red. At the external boundary, this prescribed temperature involves the information about the cooling rate of the system. The cooling rate is a key experimental parameter that can be varied, and we will be interested in understanding how different cooling rates affect cell damage. At the freezing front, the key condition is the freezing temperature. The reason I flagged it is that this is the condition that couples the heat and mass transport in the system. This condition means that the system can exhibit constitutional supercooling, and I'll talk about why this is important a little later. So this is the full mathematical system, and there are different ways in which it can be solved, each of which have their own benefits. One way is to solve it numerically. The full details of how to do this are outlined in our paper, and a similar problem was solved by Anderson, Benson and Kearsley last year, where they covered this method in more detail, so I would encourage you to read both if you're interested. Very briefly, the way we solve this system numerically is by first transforming the moving boundary domain into a fixed domain. This transformation has a cost, but the cost is sidestepped because we can solve the system using the method of lines. There are a couple of difficulties that can arise from this, outlined here, but we can sidestep them with some thought. Please do ask if you'd like to know more about this. So let's see what the solutions look like. I'm first going to show you the temperature through the system over time. On the left, we have slow cooling, and on the right, we have fast cooling. The x-axis is space, and the movie evolves over time. The dashed line is the position of the freezing front. You can see that the fast cooling has a clear spatial dependence in the temperature, whereas the slow cooling is pretty uniform throughout. This trend is a clear difference between fast and slow cooling, and this will be important when it comes to evaluating cell damage later. Now I'm going to show you the CPA concentration in the system over time. Again, we have slow cooling on the left and fast cooling on the right. This time, the dashed line is the position of the cell membrane. You can again see the spatial dependence that appears for fast cooling, which is just not present for slow cooling. This is despite the fact that both simulations end with the same solutions of CPA concentration in the system. It is the transient behaviour that is different between the two cooling rates. Now, these numerical solutions are very nice to have. They let us see what types of behaviour the system can exhibit. But they take a couple of hours to obtain, and by themselves, they don't tell us too much about what is going on in the system and why. We can get more information from the model by analysing it in a different way. The way we do this is through a mathematical technique known as an asymptotic analysis. In this technique, you exploit extreme parameter ratios in the system. That is, you're less interested in whether a particular parameter is 100 or 200, but more interested in whether that parameter is around 100 or 0.01. These sorts of extreme parameter ratios make the problem difficult to solve numerically, but helpful to analyse asymptotically. Moreover, they mean that you can obtain solutions in seconds rather than hours, which means that you can efficiently sweep through experimental parameter space using this method, as well as gaining a fundamental understanding into the physical processes involved. In this problem, we have three naturally occurring disparate timescales, which mean that we can analyse the system in this way. These timescales are heat conduction, which occurs over a few seconds, CPA diffusivity, which occurs over a couple of minutes, and cell membrane movement, which occurs over a bit less than an hour. I'm going to skip over the details of the asymptotic analysis and instead tell you what the analysis tells us about the cryopreservation process. I should note that there is one more timescale, the timescale of cooling, but this timescale is user-defined rather than natural. What this means is that the richest possible system behaviour over each natural timescale occurs when the cooling timescale coincides with that natural timescale. To avoid getting into various subcases, I'm just going to talk about the richest possible behaviour, starting with the fastest timescale. So, over the heat conduction timescale, the two moving boundaries, the freezing front and the cell membrane, are essentially static. The problem is just one of heat conduction with no further coupling, and this can be solved analytically. Over the CPA diffusion timescale, the cell membrane still doesn't move, but the freezing front does start to move into the liquid region. As it advances, it pushes the CPA out and causes a build-up of CPA close to the advancing freezing front. This slows down the movement of the interface and can cause constitutional supercooling, which I'll talk about in more detail later. Over the last timescale, you finally start to see the cell shrink. Now the system is only limited by how quickly the CPA can permeate the cell membrane, and the governing equations systematically reduced to the well-known Kedem-Kachalski equations. So now we understand what's going on in a bit more detail, and we have our efficient asymptotic solutions. We can start to investigate the problem of cell damage. If you recall, there were different damage mechanisms for fast and slow cooling, namely intracellular ice formation, which we'll assign to supercooling for reasons I'll mention shortly, and solution effects injury, which here means CPA toxicity. So we'll want to introduce different damage metrics for each, starting with intracellular ice formation. One key aspect of our model is that it only allows ice to form at the freezing front. In many cases this wouldn't be an issue, but in this problem constitutional supercooling can occur because of the coupling between freezing temperature and CPA concentration at the freezing front. This means that the temperature in the cell, away from the freezing front, can become colder than the local freezing temperature. You can see from this video that the supercooling starts at the freezing front, then propagates into the liquid and cell, getting quite high within the cell. In practice, this means that if you have nucleation points away from the freezing front, which you typically will, you will get ice forming away from the freezing front and perhaps inside the cell, causing intracellular ice formation. So while our model can't explicitly account for this intracellular ice formation, we are able to infer its effect from our model. Essentially, we need a metric that accounts for the supercooling in the cell above some safe threshold, and that is what is given by the S metric here, which is a proxy metric to quantify the damage due to intracellular ice formation. I've plotted this metric for different cooling rates, and you can see that it increases as cooling rate increases, which is what you would expect intuitively, but our model is able to quantify this. The crosses are the numerical results, and each cross takes a couple of hours to obtain, and this solid line is the asymptotic results. The whole solid line takes about 20 minutes to generate using the asymptotic results, and you can see that it agrees well and allows for a better resolution, so it really is worth using the asymptotic results. The other damage metric is for CPA toxicity, which is more prevalent at lower cooling rates. For this metric, we want to quantify the cumulative CPA exposure of the cell, accounting for the lower metabolic damage at lower temperatures, and the result is given by this T metric here. This is similar to the CPA toxicity cost functions introduced by Benson and co-workers. I've plotted this metric for different cooling rates here, and you can see that it increases as cooling rate decreases, as you'd expect intuitively, but our model is able to quantify this. Again, the asymptotic results agree very well with the numerical results and are much quicker to generate, so they are well worth using. The penultimate slide here shows how you can use these two metrics to obtain an overall combined damage metric, denoted by D. I've derived this using a linear combination where the strengths of each will depend on the susceptibility of a given cell type on each damage mechanism. I've plotted 4 different curves which correspond to 4 different resistances to supercooling. You can see that the optimal cooling rate, which is the minimum point on each curve, increases as resistance to supercooling increases, as you'd expect. But this figure represents a quantification of that in terms of the parameters of the system. So, to conclude, essentially we've derived and solved a model for single-cell cryopreservation. We solved this in different ways, which allowed us to develop physical insight into the underlying mechanisms. For example, the 3 disparate timescales and different processes occurring in each. It also meant that we could develop proxy metrics to calculate cell damage. Our solution methods mean that we can efficiently quantify and compare different cryopreservation protocols. As I'm an applied mathematician, not a practitioner, I'm sure that there are many ways this work could be extended that haven't been covered here, and I'd be delighted to chat to anyone interested in discussing this work further. Thank you for listening.